This practical exercise corresponds to the visualization and interpretation of sequence comparison using dot plots. Dot plots is a graphical method that allows the comparison of two biological sequences and identify regions of close similarity between them. We're going to use, for the dot plot analysis of the sequences, a tool that is in the XPASY site under Proteomics, Similarity Search and Alignment, .let. This tool was developed by Marco Pagni and Thomas Junier from the Swiss Institute of Bioinformatics. Let's go to describe the basic procedure for using this program. First of all, we have to click in the input button, and this opens a sequence input dialog. And in this sequence dialog, we can paste the sequence that we want to compare. In this case, we have a sequence here. And now the sequence is entered in the program. We have two windows for entering sequences. In this case, both are the same because it's the same sequence. It's the only sequence that we enter in the program. If we want to select the horizontal sequence, we select the first menu, and if we want to select the vertical sequence, we select the second menu. Now we can compute the similarity between the two sequences. In this case, it is a perfect diagonal because it's the same sequence that we are comparing. How we interpret the coloring in the displays area? Well, each pixel corresponds to a residue in the horizontal sequence and another residue in the vertical sequence. And the pixel color depends on how similar the two sequences are around these two positions. So each pixel represents a score, with high score meaning good matches. In this case, the darker the pixel, the lower the score. The good scores are here in white. Also, we could inspect the alignment at some spot on clicking on the corresponding pixel dot window. This blue cross indicates in what region of the sequence we are moving. If we approximate to the diagonal, we reach the correspondence between the sequences. Obviously, there would be a large number of pixels with low scores, and only few ones with high scores. So it is likely that you will have to tune this grayscale in order to make the background noise or low scores disappear and the similar regions stand out more clearly. For that, we can use the histogram window. The histogram window represents the frequency of each score over all the pixels on a linear in blue or logarithmic scale in purple. The lower scores are on the left and the highest on the right. So this large peak on the left corresponds to the majority of pixels with low scores. If the sequences have some similarity, as is the case, there will also be a small peak at higher scores. And this is the region that we are interested for. So we could modify the scale, getting rid of all this area here that is not informative, and focus on the region of similarity. This program also allows to use a different metrics for the comparison of the sequence. That means the use of different substitution matrices, from the identity matrix to the Blosum matrices and PAM matrices for amino acid sequences. Let's go to see some practical examples about how to use Dotlet and word interpret the result. Here we are a sequence of a receptor, a human receptor, and the sequence of a homologous receptor but in a different species. If we compute the dot plot for the comparison of these two sequences, we see clearly that could be a region here of coincidence. We can play with the histogram window to identify the regions with high scores that contribute to the similarity between sequences. Here we have the region, and we can see the diagonal, in this case for the two sequences. If the sequences are not related at all, when we compute a dot plot of two unrelated sequences, we never going to see a region of conservation between them. Let's go to compare two other sequences and see two other examples. In this case, we are comparing the sequences of two proteins of different species that share one domain in common. Here you can see that one sequence is shorter than the other. However, they contain a region that displays similarity between the two sequences. In this case, we are talking about a functional domain that is present in proteins of different size and maybe different function. In this case, This domain corresponds to the family of homeobox binding proteins. There is a very common fold that are shared by many structures for the and is used for the binding of DNA. So, in this case, we detect the position of this domain in the sequence of the vertical with related to this small sequence. Another interesting example that we can see is when we compare a sequence with itself and we see, of course, there is the clear diagonal, but in this case, if we adjust the values of the histogram, we see a region of conservation, like a pattern of conservation, in this form. What this represent? What this kind of pattern represent when you compare a sequence with itself? We can invert the grayscale to try to see more clearly. Here we are. Here is the pattern of conservation of the sequence with itself. Here we have an example of a protein that contains repeated domains. In this case, this is an example of the doublet application. When we are comparing, it's a protein that has these structural domains that are repeated, and when we compare the sequence with itself, the correspondence between domains are seen in the form of these patterns of conservation. If we observe closely, we can see that there is a region of moderate conservation between each of the domains. This is our repetitive domain, so we can see that there is a clear pattern of repetitions of leucines, and so on. This is another interesting observation that we could obtain from a dot plot application. Here we have another situation. Here we have the comparison between two proteins, and we see this pattern. What does this pattern mean? There is a perfect diagonal onto this region. As you can see, there is a perfect match of the two sequences. Then the correspondence is lost and appear in this part. So there is a displacement of the diagonal. This implies that there has been one deletion of one sequence with regard to the other. There is a stretch of amino acids that have been lost in one sequence with regard to the other, and that is why we see a displacement of the diagonal that is proportional to the number of residues that has been lost or gained by one of the two sequences. This is a classic pattern of insertion of or deletions. Well, there are more examples in the web page of .NET that you can study. This is the repeated domains. Also for the detection of exons and introns in sequences. The chain terminators, that there are palindromic sequences that they are usually represented in a dot plot as an invert diagonal, because sequences are palindromic. The low complexity regions, that there are regions of repetitions between the sequences, so they usually, you can see like a big region of a strong coincidence, but this due to the presence of the repetitions. And some other examples. You can use the application and you can play with the application to see how we can compare graphically two sequences. And that's it for this practical exercise.